Well, Dr. Jasper, you've been one of the pioneer and a witness in the development of neurosciences, and with Dr. Penfield, you've been the strength really behind the Neurological Institute in Montreal. And on behalf of the American Association of Neurosurgeons, I would like to go over you and cover some important aspect of chronological aspect of your life. And as a start, I'd like to ask you about your parents, your father, mother, your grandparents. What influence did they have on your career?
Well, I've always thought that one should try to understand one's genes in order to understand one's behavior. And so I've looked into this a bit. And on my mother's side, it was the Huguenots in France that she came from, her family, persecuted in France over the years with their beliefs in Protestant beliefs against the Catholic Church, and finally escaped with many others, thousands of them, from France. And her group went to Switzerland and hid away in the mountains of Switzerland. And from there, my grandfather had his family in a little village up in the mountains above Aigle in Switzerland, where my mother was born. And they came to the United States. My grandfather, with 9 children, migrated to the United States, and his family grew up in the prairies of the United States on Indian territory, and finally migrated to Oregon, and where they met my father, who himself was a pioneer. His great-grandfather went to Oregon with the Oregon covered wagon over the Oregon Trail, and they were one of the very first settlers, English settlers in Oregon. So that both sides of my family were, I call them restless. They were full of restless genes who make them do strange and adventurous things. And I think that sort of tendency to try to do something different has influenced my life too a great deal.
Is she the one who taught you French, by the way, your mother?
Yes, my mother was from French Switzerland, and they spoke French in the family with her parents, her sisters and brothers, and her father and mother, so that I learned to hear. I heard a lot of French at home, but my father didn't know a word of French, so we didn't speak French in the home between us. But I got used to the language, and that helped me later on in life to learn it more quickly.
Yeah, when you came to Montreal and then later on in France, I'm sure it must have been.
Well, I learned it mostly in France. I spent 2 solid years in France, living with French people, and there I by necessity had to learn it. But my father was the greatest influence on me, actually. He was a minister of the Protestant church and quite a scholar, graduated from Willamette University in religious studies. And I grew up in a very religious home with him, with a big library on comparative religions and philosophy, which I perused even at the age of 8 and 10 years of age. And this influenced me a great deal because I was very much affected by this training in religion and philosophy.
So what were your initial studies? What did you embark on originally?
It started out in philosophy at Willamette University, the same university my father graduated from much earlier. And then I went to psychology because I found philosophy, although fascinating, was a little too wordy for me. There wasn't enough concrete facts involved in it. So I finally went over to psychology instead. And I had a very excellent psychology teacher that introduced me also to the State Mental Hospital in Salem, Oregon. And incidentally, the superintendent of this hospital had a daughter who was in my class in psychology at the university. And she introduced me to the hospital. It was my first intimate contact with mental patients, which influenced me very, very much indeed. for the rest of my life. It was a fascinating experience. And from there I went to Reed College, where I did another thesis, if you like, on optimism and pessimism. It was my first publication. It was studies of college environments and what causes students to get depressed and commit suicide in college environments. And I surveyed a number of colleges in Oregon to try to solve this difficult and important problem.
What year was this about, approximately? What year was that?
This was 1927.
And what your main conclusion? This is a very, still a most
Well, I thought that there were
topic.
two big standards, conclusions that stood out, that most of the ones who got into trouble came from very religious backgrounds, very fundamentalist religion, and they couldn't face the changes in their attitudes that they learned in philosophy and psychology at the university. It was too much of a shock to their whole way of living. And then the other reason was that these students couldn't face a challenge of a difficult examination, and they crumbled under it and gave up, and those were the two big causes of difficulty.
Did you ever consider the medicine at that time? Or
No.
you were not interested?
I wasn't interested in medicine.
Why not?
I wasn't particularly fond of doctors, and I wanted to be sure.
You were fond of doctor, or you were not?
I was not fond of doctors from a childhood, and my experience was not pleasant. And I'd made up my mind, working with my father and my teachers, to give my life to brain research. I was about 16 or 18 years old when I decided on my future career, and this was in 1929, I think. I dedicated my life to brain research, and I've followed it through all my life, and have never deviated. Even, it may have seemed so, but I was going around to come back to it in the end.
Yes. But somehow you were a bit disappointed with psychology and
Right.
philosophy and
Right.
religions, and so what made you really change your mind, or to move from psychology and philosophy to physiology, for instance?
I felt as though we had to have some more direct objective measurements of brain activity, not go on speculation as the psychologists did from paper and pencil tests and that sort of thing. It wasn't adequate for me. And although I did some experimental psychology, wrote a thesis on perseveration and behavior, and it had laboratory studies in it, I wasn't satisfied with the kind of data you were able to get with psychological techniques, so I looked for something more objective, like electrophysiology, for example, which I started at Iowa.
Did you work on cerebral dominance?
That's at Iowa.
About that time, huh?
I did quite a few studies using electrophysiological methods on bilateral organization of the sensory motor systems, movements of the eyes, and speech in stutterers and normal speakers, to analyze what was the nature of cerebral dominance and its effect on speech. That was my doctorate thesis.
Did you come with any solid conclusion about this?
Yes, I thought that the evidence was quite clear that many of the stutters had a very severe disturbance in cerebral dominance, mixed dominance of eyes and hands, and in coordination, even in their eye movements. Their eye movements would stutter, right, reading a page, just like their speech would stutter. So it was a general difficulty they had in coordination of the two sides.
And who were your early mentors at that time? Main influence?
Well, I had two at Iowa. One was Seashore, the author of the musical tests of musical talent and ability, and Lee Edward Travis, who was a speech pathologist, and he began the electrophysiological studies with me.
You hadn't met Mr. Money at that time?
No, that was, well, I met him soon, because we went to Chicago for the physiology meetings, American Physiological Society, and he came with his colleagues from St. Louis, Missouri, Erlanger, Gasser, and Bishop, who were doing the first studies with the cathode ray oscillograph of analyzing the nerve action potential. And I met him in Chicago, and I liked the cut of his jib. He was a fine young fellow and very able. And we agreed that he agreed to take me on in his laboratory in Paris, in the Sorbonne.
Was he already working in France, or
Well, yes.
he was working in...
Sure, he had his doctorate in Paris before.
So he was just traveling to the States?
He was a postdoctoral fellow of the Rockefeller Foundation, studying in St. Louis just for a year. So that since he was a Rockefeller fellow, he got in touch with the Rockefeller Foundation and managed to get me another fellowship to go over to France and join him at the Sorbonne, where we spent 2 years of very productive work.
Yeah. So what year was this?
That was 31.
1931, and this was to be two of the great years in your life, I suppose, working in Paris and in your surrounding. And of course, as you mentioned earlier, you were fairly fluent in French at that time, were you?
Not very.
Not very.
No. I had to learn it. I had a tutor in Paris to help me learn it, and then I lived in a French family. In fact, I lived with the Monniers. They took me into their home, and we spoke French in the home all the time. And then I spoke French in the laboratory. Got so I could give papers to the Société de Biologie in French, and this helped me.
And the big boss at that time in La Sorbonne was Professor Louis Lapic?
Louis Lapic was the chief of physiology, yes.
How was he like? He was the Chronaxi man?
Yes, he was a very dominant personality, but a kindly man, nevertheless. But he had this theory of chronaxy of the nervous system, which was fascinating. I'd read about it before, and I'd actually done some chronaxy measurements in Iowa after reading his work. And I found some very interesting results. The change in the chronaxes with alcoholic intoxication, for example, and difference in the two sides in right and left-handed people. So I was interested in continuing to do something with this measurement of the time constant of nerve action potentials. So I did some with Monnier, and we finally found that it wasn't quite as clear as it appeared in Lapic's books. And we wrote a paper with Monnier on the pseudo-chronaxes, measurements in crustacean, which was a criticism of the method. And then gradually we realized that there was a good many flaws in the theory. A very interesting theory, though. I still am interested in it, but it doesn't apply now to what we know. It was the idea that transmission of the nerve impulses across synapses depended upon a tuning, electrical tuning, of the time constant of the presynaptic fiber and the receptor. They had to be in tune, timed, like you'd tune a radio program, get the right frequency.
Very more modern concept.
I thought it was a fascinating idea. This was before we knew about chemical transmission of the nerve impulse.
PSP and IPSP and all these things were not discovered. Did he put you to work on a thesis? Did you register for a thesis work at that time, or? I
didn't until I'd worked a year, and then I was getting so much interesting results. I said, well, I better just apply for a thesis. I might as well. I didn't plan to do that originally.
And working on crustacean?
I was working on the crustacean nervous system and the effect of the ganglion on the peripheral system and the excitability of nerves and muscles. It was the same problem. What does the brain have to do with nerve muscle excitability? And in the crustacean, it was very easy to study. And we did a lot of work on the lobsters and crabs and things like that at the Roscoff Marine Biological Station.
This was way out, was in Brittany, huh? Roscoff?
That's in Brittany, yes.
Did you spend the summers there?
2 summers.
Yeah. Sailing?
But we did sail. But it was the most, one of the many experiences I had which are most fruitful, in that it was a mixture of molecular biologists, men working on genetics, men working on chemistry of nerve, like the Krebs with the Krebs cycle was there. And Jacques Monod,
Uh-huh.
the molecular biologist, became a very close friend.
Both Nobel Prizes?
And they're both Nobel Prize winners. Eventually, they weren't at the time, of course. But Monod was my chess partner. We played chess together every night. And then I found that he was a sailor, so we joined in sailing in the British Channel, together with Lapique.
Everybody had a sailboat then.
Lapique had
with fancy neurological names.
That's right. Lapique's boat was named the Axone, and it was a big ocean-going sloop. It required a crew of 2 Breton sailors to sail it out of the
Across
harbor.
the channel?
And across the channel we went across to Plymouth, where there's another biological station in England, and went over there to see what they were doing, and then we sailed back to Roscoff in his boat. Had a little trouble now and then. We ran on a reef once getting in, because they have huge tides there, 30-foot tides, and we thought we could make it before on this channel, and the tide went out too fast and let us sit on the reef there. But we managed to get off.
And Al Monnier, too, I know he was a very dear person to you.
He had a sailboat.
Well, he also had a sailboat.
Well, he sailed with me in the Lapiques. We went on sailing together with them, but then he got a boat, and we sailed later on his boat, too.
Yeah.
We even sailed in the Seine River.
Yeah. And in those years, did you hear about Hans Berger and the development of electroencephalography and electroencephalogram in man?
Well, Berger had started his work in '31 when I was there, but he wasn't well known. And the German research workers in the Sorbonne who knew of his work told me that he was a small town psychiatrist in Jena and had no experience in electrophysiology, and probably it isn't worth going to see him.
So, essentially at that time, these 2 years they were spent in La Sorbonne and starting on your thesis, and then eventually you returned to United States.
In '33.
In '33.
Yeah.
And where did you go then?
Went to Brown University at Providence, Rhode Island, and the Rockefeller continued to support me, and we built up a big neurophysiology laboratories at Brown in one of the hospitals, and it's there that we started to study the electroencephalogram. Actually, I heard more about it through Adrian, who came to the United States to the meeting of the American Neurological Association in Atlantic City. They invited him to give a lecture on brainwaves. He came in '34. I went to the meeting, and I was very much impressed. And we went right back to our labs and started working immediately.
And Adrien, was he trying to reproduce, in a sense, the work of Berger, or this is original work?
Yeah, he was confirming first before he went to do other things. He of course, went further and showed some very important things himself in regard to the effect of attention and psychological processes on the brainwaves. He discovered that himself sort of by accident. And,
but I suppose your competitors to be in the United States, they must have been around, and they must have heard
at
of his work too.
At Harvard, there was Alex Forbes, Hallowell Davis, and Gibbs and Lennox were working there very fast to try to beat me at the development of the EEG clinically. And I was down just 50 miles to the south and in Providence. And we had keen competition to see who could have the first publication. And we won.
You won? Who were your main collaborator at that time?
Well, the main important one was Dr. Andrews, who was an electronic engineer and physicist from Brown, who built all our apparatus. And he also did some of the experiments with us. Couldn't have got along without him. And then the chief of psychology, Dr. Carmichael, joined me in the psychological studies. And another neurophysiologist from Yale joined me later, Reinberger.
Yeah, okay.
So I had a good team.
And after a while, I think you went back to Europe, and you had to present or defend your thesis.
Yes.
And by that time, you had 2 theses, if I remember, to defend.
In order to finish the work on crustacea for my thesis in Paris, I went to the Marine Biological Station at Woods Hole, which was near Providence, Summers. And I completed the data that I wanted, wrote the thesis. I wrote it in English, and Madame Monnier in Paris translated it into French for me. Very nice of her. And presented it to the Sorbonne. And then I went back in 35 to defend my thesis at the Sorbonne. And I wrote a second thesis on the electroencephalogram.
And at that time, you managed to finally go and see Hans Berger.
Yes.
I think you were able to retrieve him and visit him. Do you remember him?
Oh, very well. Yes, as soon as I finished my defense of my doctorate in Paris, I immediately went to Jena to visit Berger. Of course, I visited Fessard, too, in Paris, who had done some EEG work.
Not Madame, Monsieur
No,
Fessard.
Alfred Fessard. He was the most distinguished neurophysiologist in France at the time, and he worked on the EEG with some colleagues. The Dean of Medicine, Baudouin, there was in on it, and Henry Fishgold, so I got some good contacts in Paris with those interested in EEG. Then I immediately went to Jena to visit Berger, Hans Berger, and he gave me a very nice reception in his home with his family and for dinner, and showed me all his records in his hospital and so forth. And I had a pocket full of records I brought with me to show him, and fortunately mine were taken with a mirror oscillograph, and they looked very much like his. He used a Siemens mirror oscillograph, similar equipment. And so I said, well, now these, I can show you that these really are brain waves, they're not artifact. And he was so happy to see that somebody else with the same equipment could get good records, because his colleagues in Germany were saying that, oh, well, his records are probably artifact. And of course, he'd been convinced also by Adrian that his records were genuine. And we had a wonderful time. He showed me his lecture theater in the University of Jena. And back of his lectern was a great big picture of Hitler, Adolf Hitler.
Yeah, that was just the pre-war
35.
period.
And but the Nazis were very active in 35.
Yeah, sure.
And he was instructed to have his class stand and click their heels and say Heil Hitler every time before he started his lecture, because of the picture, you see. He refused to do this. He thought this was ridiculous, and he didn't like the Nazis anyway. So this got him into trouble with the Nazis right from then on, and serious trouble. And finally, during the war, he was so shocked by the treatment of the Jews and what was happening to his good fatherland that he admired so much, he went into a deep depression. He was taken out by the Nazis from his laboratory and from his university post. He retired to a house in the country, but finally got so depressed he took his own life during the second year of the war.
And you went on and I think visit also the folk,
Yes, I went from
Oscar Folk and Cecil.
Yeah, I went from Jena to Berlin, and the Folks Laboratory was a suburb of Berlin called Buch, B-U-C-H, Buch by Berlin. It was the Brain Research Institute, a very big place. The Cecile and Oscar Vogt had created this with a large number of colleagues, among whom was Dr. Tönnies and Dr. Kornmuller, who were doing electrophysiological work on the brain, electrocorticograms. They were studying the cortex of monkeys and cats.
The correlation, the electrophysiological correlation of the histological parcellation or various areas.
One of the Vogt's great contributions is to devise a cytoarchitectonic map of the brain. They divided the cortex into about 200 areas from the microscopic observations of the organization of the cells and fibers in these as they changed from one part to the other. And he drawn maps of this, And Kornmuller thought he could detect from the brain waves every time he crossed one of Volk's boundaries into another area of cortex, he would say, oh, well, that's a different pattern, you see. So he was able to support Volk in all his cytoarchitectonics. But of course, it wasn't all easy for other people to see. I mean, he only was able to see some of these changes as you crossed from one to another. But there was a good deal of truth in it, because later I was able to confirm quite a bit of that, recording from the cortex, that the pattern from the precentral gyrus, the motor area, was quite different from the postcentral, although some similarities. And as you crossed into the parietal lobe, you begin to get the big alpha wave pictures, another pattern in the occipital lobe. There were these very clear differences in different parts of the brain.
The different substratum from various regions.
Yeah.
Quite most interesting. Olszewski was not yet working with folks, nor Hassler, huh? Rolf Hassler. You didn't meet
They weren't there in 35.
It was too early.
They came later. After the war. Olszewski and Klatzo came
later on.
from as Polish refugees to the Volks. And that was when the Volks were put out of their lab by the Nazis in Berlin and established a laboratory in the Black Forest. And they had a special protected laboratory there so the Nazis couldn't bother them. It was because they were great friends of some of the manufacturers of munitions in Germany, the Krupp family. And the Krupp family told Hitler he'd have to leave them alone, and he built them this laboratory.
So this gave you another nice opportunity to meet most of the neurophysiologists of the time in Europe at that time.
Yes, and we went to many meetings all over Europe, and clear from Italy right through to Switzerland, France, and Belgium, and Denmark. Saw physiologists throughout there.
You had your both thesis behind you, and you came back to the States again.
Well, before I came back, I went to England to visit Adrian and Matthews and Gala and Gray Walter, who had EEG labs going there. I visited their labs, and we had a great time. And then we went to Oxford for the meeting of the English-British Physiological Society. And it was a very famous meeting. It was the meeting that Sherrington gave what they call his swan song, his resignation, his retirement from physiology. And he gave a wonderful address at the time. We were meeting in honor of Sherrington at the time. And I met Jack Eccles and many other Oxford people, A.V. Hill and so forth, while I was there. And then I set sail back to Brown to complete my work there.
And then at that time, I think it was you or Dr. Carmichael that I had heard about some work being done in neurosurgery by Dr. Penfield up in Montreal.
Right.
And you invited him or something?
Yes, we invited him to come down and give us a seminar at Brown in the psychology department. He came, gave us one of his fascinating talks with beautiful colored pictures of the brain surface and all the tickets where he'd found responses from electrical stimulation. And then he talked also about his ideas of consciousness and how consciousness was organized in the brain. He'd already written a paper on this subject before. He gave a Harvey lecture on this. So this interested the psychologists and the psychiatrists very much indeed.
And yourself particularly, because this is what you've already had always been interested in,
exactly.
Right from the start.
This fascinated me because it was the mechanisms of consciousness and mental activity related to brain activity which is driving me throughout my life.
Yes.
And so I asked Penfield to come out to my laboratory in the hospital and see what we were doing, and he came, and I showed him the recordings and how we did it, and then I showed him pictures of records of 2 epileptic patients I'd examined and localized the focus of epileptic discharge. And he said, "You mean you did that from those wires just stuck on the head?" I said, "Yes, that's the way we do it. If we have the proper apparatus, we can do it that way." And he said, "Well, that seems very unreal to me. I don't. You'd have to show me that you really have a focus with that sort of method. I said, well, do you mean that? Do you mean I'd have to show you? He said, yes. He said, well, what if I bring my patients up to you? Will you operate on them and see if I'm right?
Yeah. So he knew a lot about the stimulation, but very little about the electroencephalography, in fact, or brain recording.
The electroencephalogram. He didn't understand it at all at the time.
This is very typical of the early epilepsy neurosurgeons.
The Gibbs had already worked on epilepsy in Boston, but he hadn't followed their work. So,
I think it was very typical of many early neurosurgeons involved in epilepsy. You know, they were looking for, at that time, they were looking for a lesion or a scar, and not too interested in the electroencephalogram. But you really changed his mind, and whatever happened of these two patients?
Well, we brought the patients to the Neurological Institute, and then my engineer built me a portable EEG apparatus with pens, 4 pens on it. And
four channels. And
4-channel pens, pen recorder.
These were original amplifiers?
Well, no. He built the amplifiers. We bought the pen recorder from Offner in Chicago. They were ready-made ones, crystallographs. And we put those in the back of the car, and I drove to Montreal on weekends and commuted for a year with Penfield doing the operations and me recording in the operating room.
On the exposed
cortex, yeah. We built the electrodes for that with Dr. Cipriani at the Montreal.
Dr. Cipriani, he was
the electromechanical
engineer
at the Montreal Neurological. And he joined me when I came up there and helped us build all the equipment we had there. He built the pens and amplifiers and everything, all with his own hands.
And were you able to get decent tracing, or did it take a while before you?
The first one was a great disappointment because
Mr. Carrozza.
It was full of 60-cycle artifact from all the apparatus that was in the operating room. Was broadcasting such a powerful error artifact that we couldn't get rid of it the first operation. The second one, we managed to get rid of the artifact in the operating room. So I set up and we were recording very nicely. And then pretty soon, Dr. Penfield heard some curious music. And he said, where did that come from? You know, he takes off his glasses. Where did that come from? And we looked over there, and it was coming from the EEG machine. The EEG machine was picking up the radio tower on the top of Mount Royal, and we got the radio program together with the brainwaves. So I did have my problems at first.
Yeah.
We finally solved them.
So, but then you were not yet part of the institute. You had to commute for
a trial period.
Yeah. Then they managed to change your mind, and you finally came to the Neurological Institute.
Yes.
What year was this?
38.
1938.
38.
And who did you meet up here in Montreal?
Well, of course, I'd got acquainted by my computing trips with the staff and was very much impressed with all the fellows and the surgeons and the life. On weekends, we'd always go skiing, the whole bunch of us, the staff and the fellows. We had a ski cottage up in the north, and this appealed to me. And the fact that the nurse who was in charge of Sammy Carosa was a very efficient nurse and a very good tennis player, and I got interested in her too. So there were other reasons for my wanting to come to Montreal.
She eventually became your first wife.
That's right.
Yeah.
That's right. But Penfield and I got together on a plan to bring me to Montreal because he knew the then president of the Rockefeller Foundation, Alan Gregg, who had given him a lot of money to build the Neurological Institute. And he got in touch with him and said that he wanted to bring me up to Montreal, but the institute was finished and there was no room for my laboratories. He would need an additional grant to be able to build an annex to the institute in order to accommodate the EEG laboratories. Well, Rockefeller came across with half of the money for this new building, But he said he'd have to raise the other half in Montreal, a matching grant, which Penfield was able to do through, partly through Mr. McConnell, the McConnell Wing, and the city of Montreal gave some money to it and so forth. They made up the difference. So they built an annex, and that's where I came to open formally in 1939, in February.
The Department of Clinical Neurophysiology and Electrophysiology.
That's right.
But before that, there were already people interested in neurophysiology and neurosurgeon, and I think Dr. Ted Erickson was already...
Erickson was interested in Arthur Elvidge. Elvidge was technically in addition to being a neurosurgeon, was head of the neurophysiology laboratories when I arrived. And he had a PhD in physiology in addition to his doctorate. So they were interested in...
Bill Cohn was there too?
Bill Cohn was there, very fascinated with the idea of doing electrical recording, but he didn't know anything about it, and he spent so much time in the operating rooms and on the wards that I really didn't see him very often, excepting about midnight, when we'd be doing an experiment in the wards. He'd be dressing the wounds on some of his patients, and we'd have coffee together at about midnight.
And Ted Erickson, was he a fellow in neurosurgery at that time?
Yeah, resident. And he became a staff member afterwards, and he published the first book that Penfield wrote on epilepsy, was Penfield and Erickson, called Epilepsy and Cerebral Localization. And I wrote a big chapter in that book. The first chapter I wrote on the work with Penfield and the EEG.
But you were working on the spread of seizures, and he was working already on the corpus callosum.
Yes, Erickson and I did a study in animals, doing experimental epilepsy in animals, to analyze the manner of spread from a focus. And we created artificial foci and then studied the electrical spread of the focus from one hemisphere to the other across the corpus callosum and finally to subcortical areas.
I suppose the most, maybe the most impressive person at the time was Wilder Penfield himself. Maybe this is a nice opportunity to tell us, how he impressed you and what you thought of the man himself.
Penfield and I had so many common interests in the study of the brain. I found that he wasn't primarily interested in epilepsy. He was interested in what epilepsy could teach him about the brain. He used his epileptic patients to teach him about the organization of the brain. Well, this appealed to me very much because that's the way I work too. And we were very glad if we could help treat epileptics, but our main interest was to study how the brain worked. And this was a great opportunity. And so we got together right from the beginning. And then, of course, he was a keen sailor and a skier, so that we got together outside of the laboratory as well. And I joined his family in Lake Memphremagog for sailing, and then we skied in the winter. So we became very close friends. He sort of took me into the Penfield family, and later on became the godfather of my son. But of course, our most important observations at that time were the ones made from the surface of the human cortex exposed at operation. And he would operate once or twice a week, and then we'd be studying these records together in epileptic conferences. So this was the most productive collaboration I had with Dr. Penfield. Now in the EEG lab, it was Dr. Kirschman I collaborated with. He was a very bright neuropathologist and neurologist who became very much interested in electroencephalography and did my first big study of EEG classification of the epilepsies.
Tell me about this classification. Was this based on just purely electrographic patterns, or?
No, that was only one thing we studied, was the EEG, and we always studied at least two recordings of the EEG, and whenever possible, to record a seizure. But each patient was studied from a clinical point of view with a careful clinical history, observation of seizures, X-rays on all of them to rule out organic changes in the pneumograms. And in certain cases, we did special metabolic studies. So every patient got a thorough workup of this sort, as well as the EEG. In order to study EEG classification, you have to see what the EEG, how it fits into the rest of the clinical picture. And that's what we did.
And there was already quite the spirit, quite a family within the neuro at that time. There was a lot of fellows in neurosurgery and
the unique
EEG
thing about the neuro was the spirit, and Penfield's dream of creating an institution where research and clinical work were combined as in teamwork throughout, and where all the different disciplines of neuroscience were represented: anatomy, physiology, chemistry, neuropsychology, all neuropathology. And all of these disciplines were joined in teamwork. And we often met together, all of us from the various laboratories, studying clinical patients. So it was the first big institute that combined all neuroscience disciplines around a clinical program.
In fact, many of the neurosurgeons and the fellows in neurosurgery or resident in neurosurgery went through your lab over the years and did thesis and PhD work. And
well, I learned to have high respect for neurosurgeons in the neurophysiology laboratories. They were very good neurophysiologists because of their excellent techniques and their objective approach, and they soon learned the electrical methods of recording. So that I got some of my best colleagues were neurosurgeons in the laboratory.
There were so many that it's almost difficult to enumerate them. There were
during the 25 years I was there, I had 40 neurosurgeons in my neurophysiology and EEG laboratories.
I have a list of them that I went through, and it's very astonishing, you know. I see, for instance, George Austin, Claude Bertrand, Gilles Bertrand, John Blundell, Charlie Branch, Charles Cure, Frank Eklund.
Yes.
Remember Frank Eklund?
Yeah, he was one of the first fellows that studied with me.
Yeah, it's the discoverer of the vasospasm.
Yeah. He
Ted,
did that in our neurophysiology laboratories.
Did he?
Yeah.
Ted Erickson.
Yeah.
Bill Feindel, Bill Gerber, Alain Godin, Jean Geibels, or later.
Jan Geibels from Belgium.
Belgium, was to work on Parkinson. Fouad Haddad, among others.
In Beirut.
John Hanbury.
Right.
Jules Hardy. John Hunter.
Yes.
John
from
Hunter.
Australia.
This is interesting because I think with Hunter you started working on one of the very first stereotaxic apparatus.
Yes.
You did some electrophysiological work with him, but also...
Well, he and I designed the first human electrostereotaxic machine at the Neuro, and it made in our own shops.
Yeah, I remember seeing a big machine built around 1948, 49.
Yeah.
Not quite comfortable, but the idea was there. And Ellis Skinner, Robert Knighton, Christian Christiansen.
Right, from Oslo.
Cholu Li.
Yes, from China. Yeah.
Gabriel Mazar
from Paris.
Paris, and Jean Panet Raymond. He was a local
neurosurgeon, Montreal.
Fano Perrault.
Perrault from Carolina.
Yeah. Well,
Charleston.
Yeah. Bernard Pertuisset from France.
From Paris, yeah.
Ted Rasmussen was. Did he work as a fellow, Ted Rasmussen?
He worked as a fellow with me at first.
Was he a good student?
Then he was a neuro. Yes, he was a student. He was a fellow as a student in electroencephalography and stimulation of the brain. But he left for the war and came back after the war to Chicago, and then finally came to us in Montreal.
Yeah. I see many others, Richard Rovitt, Alan Rudballer, Mark Report, Julius Stoll, Joe Stratford, Herbert Sylvanus, Antoine Interazzi, John Van Buren, Bryce Weir, Kesley Welsh, Andrew Wong, and many others. I'm sure that
those were fine.
Maybe I should do the same for the a very large number of fellows that you had also during that period of time, you know.
Well, you can't read all those names.
Well, I think I can, because I think it's very interesting. We could just, there was Drew Glover Fortine.
That's a selection.
Yeah.
There were 200 fellows worked in neurophysiology and EEG in 25 years.
Yeah.
And I just made a list of some of the most important ones.
Yeah, and some of them are just outstanding people. You had Drew Glover Fortine.
Yes, from Amsterdam, yes.
He was a neuroanatomist. He worked with the very early on. He was interested in intralaminar nuclei.
Yes, he was an expert in thalamic anatomy.
Yeah.
Helped us with our stereotaxic works tremendously.
There's another expert on the thalamus anatomy, Jerzy Olszewski.
Yes, George Olszewski was a German cytoarchitectonic anatomist.
Came with Klatzo.
Who had been with the folks, and came with Igor Klatzo to the Neuro as refugees from an internment camp in Germany. And they were some of our most important fellows in those days. Right. Igor in pathology and Olszewski in neuroanatomy. And he did some of the most beautiful cytoarchitectonic anatomy that's ever been done in the monkey and the human.
Yeah, the rabbit too.
And the rabbit. He wrote an atlas for the monkey, with stereotaxic atlas for the monkey. He helped us with our atlas of the cat, which we published with Imoni Marsan, which has been a very fundamental guide to stereotaxic work and cat work for all over the world. And Olszewski and Drew Glover Fortine helped us with that.
So one of these fellows also were Donald Tower,
Donald Tower, yes.
Herman Flanagan,
right.
Cosimo Ajmone Marsan, with
yes.
Henri Gasteau.
Henry Gasteau, yes. He came over from Marseille. And he spent a year with us here, with Penfield and us, the rest of us.
Yeah, I see John Van Buren also,
yes.
is one distinguished neurosurgeon. Kada from Sweden.
Birger Kada was from Oslo, Norway. And
he was very interested, if I remember, on the hippocampus, amygdala, limbic structures, and epilepsy.
That was his special field.
Yeah.
And he had an opportunity to study the limbic system in man here. Yeah. And he was delighted with that. He went to the operating room with me, and we recorded, while Penfield was stimulating the amygdala, we recorded the responses. Not only the electrical responses, the behavioral responses. The patient would stop breathing. This scared us, this frightened us, because you stimulate the amygdala and you have arrest of respiration for quite some time.
Oh, there's a very large number of them. Gilles Bertrand was a fellow, I think, working on the supplementary motor area connections.
That's right.
David Ingvar.
Yes.
Pierre Glore, your successor. David Ubel, which he was one of your fellows?
Yes, David Ubel was my fellow, my student at first, who I put through medical school by being my electronic engineer in the EEG department. And then we got interested in neurology, working around there, and he decided to go through medicine and become a neurologist. He went through the neurology training course here at the Neuro and went to the States, and I got him a job in Washington, where he did his Army service in the U.S. Army at the Army Medical Research Station at Walter Reed Hospital in Washington. And it was there that he started working on the microelectrode work.
And went on to get the Nobel Prize.
Went on to get the Nobel Prize for his study of the visual system. So he's my only Nobel laureate student.
The CIMARC report, Charles Branch, June Wada, Robert Naquet, Gianfranco Ricci.
Yes.
With Ricci,
Ricci came from Rome, and he was very important for us in that he joined in developing the microelectrode technique.
In the free moving animal?
In an anesthetized, freely moving animal. We developed a method whereby we could place a controller, a microtome, in the skull with a microelectrode in it, and we'd set the monkeys up in chairs so that they wouldn't be able to run around freely. And we recorded from single cells in the cortex during learning, conditioning. And we had some cases with Parkinsonian tremor developed after lesions in the brainstem that Dr. Poirier showed us how to do. So that this technique was used for lots of important work after this. But Ricci and I and another fellow by the name of Doan developed the method, which then was picked up by people all over the world.
All over the world.
Yeah.
Actually, one of your fellow was Jean-Pierre Cordeau, huh?
Yes. He was with us as fellows during the development of this microelectrode method, and he used it for studying experimental Parkinsonism in monkeys.
He was working with Louis Poirier, who
Louis Poirier showed us how to make the lesions for the tremor, and he came over to help us do the recording. And there was another chap from Belgium, Jan.
Geibel.
Yes,
Geibel.
Jan Geibels, who did his doctorate thesis.
Was making the bridge between McGill University and University of Montreal.
Yes. No, Geibels was with us before I went to the University of Montreal.
I know you went to University of Montreal, but the topic was very dear to University of Montreal at that time. Experimental tremor and Parkinson's disease and Dr. Poirier.
Yeah, that's right.
And so eventually you came to a point where you had to take a decision of leaving McGill and going to University of Montreal. This was an association with Dr. Cordeau.
Yes.
Tell me.
Yes, Dr. Cordeau.
Must have been a difficult decision for you at that time.
Very difficult. That was a very painful choice. But
you never regret it, I think, because you went on to...
No, we've had a wonderful time at the University of Montreal since there. And now I've been 25 years at the University of Montreal, and I was only 27 or 8 at McGill, so I've been almost as long at the University of Montreal.
Well, but every week you're still at McGill, and you're still attending rounds every week, I think. So
I still work.
I think you never left completely.
I still work at the Neurological Institute. Even the first year I went to the University of Montreal, I came back every week to work with Gilles Bertrand in the operating room, doing microelectrode studies of the thalamus in human patients.
Yeah, this was a very productive, very interesting period, I think, when this the study of micro unit, thalamic unit at that time.
Yes, we developed a special technique for microelectrode recording in the thalamus with a probe attached to a stereotaxic instrument, but it was a probe with a microelectrode curved.
Searching electrode.
A curved electrode that came out the end of our probe, of our needle, so that we could let it come out, and we'd bring it in and turn the needle, come out again, and we could explore the whole area around the insertion of the needle, which no one else had a method like that.
Not even Madame Fassard?
No, she used a single microelectrode, so that every time they wanted to move it, they had to take it out and put it in again.
Yeah. They were your competitors across the ocean.
That's right.
Guyot and Fassard, and you and Gilles Bertrand.
Yeah, she was working on my thalamic single-cell recording with microelectrodes the same time we were with Bertrand. Yeah. She was working with Guyot.
And while I think about it, we're going to have this meeting of the IBRO this coming August. I think, when was it was founded? It was before you went to...
Before I went to the University of Montreal, because the studies we did on conditioning with the microelectrode technique in animals who were awake and learning to withdraw hands and stimulations of various kinds, we were recording all these responses with microelectrodes in various brain areas, and did a complete study of the establishment of conditioned reflex in terms of the unit firing from the frontal, sensory, motor, parietal, and occipital cortex. And we had a beautiful picture of what cells do in the cortex when you establish a conditioned reflex. Well, they organized a big symposium in Moscow they called it electroencephalography of higher nervous activity, which meant for them conditioned reflexes. And they asked us to come over and present this material at the symposium, which we did. And at the end of the symposium, we were also excited with the many interesting things that being presented, plus first contact with the Russian physiologists, that at the end of our symposium, we unanimously voted to create a brain research organization, to go to UNESCO and see if they'd help us create an international brain research organization that would include all countries and the leading workers in all countries. So we went. They appointed Alfred Fessard and myself to go to Paris and present that to UNESCO, and they accepted it with great interest, because we found had previously thought of establishing institutes of research on the brain under UNESCO auspices, so we gave them an opportunity to do something about that. Well, we formed, to take a long story short, we formed the International Brain Research Organization called EBRO, and I was elected its first executive secretary to get the Constitution and statutes done, and they were done in Montreal. Yeah. And the incorporation was held in the Parliament of Canada in Ottawa.
This was 63 around that time, or
60.
60.
And 61 was the final incorporation.
And now this summer you're going to have the 1991 meeting, and yes, I think you're 27,000 members now.
The International Congress, this is the third International Congress of EBRO, is coming to Montreal this summer in August.
You're going to see a lot of old friends.
It's grown from our first EBRO which had 500 members. It's now grown to 27,000 members all over the world.
Incredible.
Terrific organization.
Time is running short. I just wanted to, for our American colleagues would be interesting to see that the NIH at the beginning, when it was formed, there was a big, strong borrowing from your fellows and neurosurgeons from the Institute.
We enjoyed a nice association with the NIH from the very beginning. In fact, when I was in Brown, Bob Felix was studying EG with me there, and he had a bunch of blueprints planning the first building for the National Institutes of Mental Health.
And who went there at the very beginning?
And the group, when the building was finished and they were looking for staff, they came to the Neurological Institute in Montreal, and we gave them a whole group of
Milton
people.
Baldwin?
The names I wrote down. You want to?
Down Tower?
Yeah. Well,
Maslan?
I wrote the neurosurgeons and so forth.
Yeah, well.
You want to read their names because it was quite interesting.
Well, I think we were running, unfortunately, we're running short of time.
We had several neurosurgeons, Mate Baldwin and Joe Lee and John Van Buren.
Yeah.
We had some neuropathologists, Glatzo and Decaban. We had neurophysiologists, were Cosimo Aimoni Marsan, Joe Lee.
William Bill Cavanis, Anthony Gorman, Costa Stefanis.
They were all in the labs of the NIH.
Donald Tower, who was
and then in neurochemistry, Donald Tower went down in neurochemistry to become
the director
of the NIH.
Like Richard Maslin was originally from
Dick Maslin also was a student with us here, and he went down to become chief. So we gave a lot of our fellows to the NIH, and we were very pleased about that.
Just tell me in very few words, how you feel, reaching the age of 85, you're still very young at heart, very dynamic. You live, tell me a little bit about the Montreal Neurological Institute and the life of neurology and neurosciences around the world.
Well, I've never left the Neurological Institute, as you know. Even though I've retired for many years, I come back and work every week at the Neurological Institute at the research seminars and lectures. And tomorrow we're having a big meeting of neurosurgeons, which
that's
I'm going to. And so this has been a great pleasure to see the development of things, which has grown very much at the Neurological Institute, new wing, the Penfield wing, and the what's the wing, the other one, Timmins.
Webster
Pavilion and the Timmins Pavilion. these are all added a lot to the Institute, but I find I'm a little disappointed in some parts of this development in that I miss the close teamwork we had in the early days.
Of clinic and basic science.
Where we had Penfield was the chief, so we called him the chief, who was interested in all the clinical and research laboratories. And we brought us all together. And our ward rounds Monday morning included the research labs as well as the neurologists and the neurosurgeons and the psychologists. Yeah. That spirit
I think this is
of
still
teamwork
very, very important.
I don't see today at the Neurological Institute, and that disappoints me. And I'm figuring on how in the world we could try to bring some of it back again, and I'm busy working on that.
Yeah. And you're, as I said, you're a very dynamic, very happy man. I think you lost your first wife, and I think you remarried, and this, I think, must have something to do with your present drive. No
I
doubt.
think that's the reason I'm not so old as I could be, is that I married a young wife who's only 49 years old now, and she's busy with the registrar's office at McGill. She's got a big job of her own, running convocations and so forth this week. But she's a young, active, vibrant person who is really
keeping you
the secret
on your
of
toes.
my happiness right now.
Okay. Well, thank you very much, Dr. Jasper.
Enjoyed it.
It's been great talking to you.